was about to ask that question and I moved on. So I'm going to go back to Amar's question. All right. So I'm going to pose it to you this way. Let's say you, Amir, are about to introduce a new purification technology into your process development environment. What are you worried about in that when you make that move? What risks might you face by the introduction of a new
purification process?
Yeah. Yep.
Well, it depends. What do we mean of a new purification process? If you're talking going fully exotic, right? Or are we actually talking about, for example, going from batch to continuous, right? That we need to have that. I think what I have seen as essential is that at the very early development, we have a very good characterization of our process. And that doesn't matter actually which direction that you want to go in order to understand, well, at what condition and in the design space that you're dealing with, you're able actually to get the most out of your purification process. So the high throughput production and high throughput purification are essential to understand that. Two is that actually I try to benefit from the tools around me. These days, there are a lot of talks around machine learning, AI, right? And to be able to use more contextualized data in order to process them and link the data that we have to final product quality attributes that and final drug substance specifications that we want to get there, right? So I don't know what specific DSP steps that this expert has in mind, right? Depending on the step, you need to have a specific measure for that. But often, I like to see change, but often actually on DSP, we're talking about filtration or chromatography steps, right? And we have a lot of means there to do that. So if I'm in a development lab and I receive a new process, I try to assess basically the fit of the ranges of the process that I have to my existing setup that I have, and try to actually to see within what design space I'm able to optimize that process. I don't think I'm answering completely the question because it's a bit also vague, right?
Yeah. Need more context.
Yeah.
All right. Maybe one last one. And this may be the last one, maybe not, because this might be a yes or no question. Kirsten would like to know if you have any experience using Deveron 16 as a virus inactivation agent. And if you do you have any concerns? No? Okay. So we'll move on. Squeeze one more in from Lillian. Lillian, perhaps, could you please talk a bit about the use of multiple column systems, especially in series, to achieve continuous bioprocessing?
Yeah, I think this is yeah, sure. I think this is something that, but regards to, as I said, bio-SMBs are actually a good example of basically those multi-column chromatographies. And what I'm more amazed of is our technologies that do not per se require huge level of complexities, and yet with existing structure, infrastructure, you're able to achieve comparable performance. What I mean is, for example, technologies to overload the column and get the 808 and reload it to the inlet, as opposed to actually applying a lot of columns on back to back. So, I mean, connected processing is something that it is in this industry for the past 15, 20 years, right? A lot of people are actually using that and are applying it. And, well, it actually saves time, right? And brings a lot of operational agility there. I think the key part is when we deal with products that are unstable, and they need to be processed very quickly, right? And there are the areas that we can benefit from a low footprint, high throughput multi-column chromatography system. So and that is the part that, well, not necessarily still you need to go for continuous chromatography. I've seen that people actually work overnight and use a column over and over with larger columns, which not necessarily is super intelligent. There are alternatives out in the market that you can still apply higher flows to the similar systems. I mean, there are products from Cytiva and JSR out there that can fulfill this. And in that way, you're able to also process your system much faster in the existing setup. That's the most interesting, right? Because you invest less, right, or zero or minimal, as opposed to actually investing a couple of millions to have a continuous system in place.
Yeah. Dr. Goudarzi, I want to do two things, right? Well, I want to do three things. One, I'm going to let you off the hook because it's late. It's after hours in North Rhine-Westphalia, auf Deutsch in Germany. It's late.
Deutschland, yes.
This is the second night in a row that you've worked overtime on my behalf, so I'm going to let you go. But before I do, I want to tell you that I want to apologize to you for putting you on the spot, completely throwing out the outline we discussed and focusing on our Q&A from our audience. But I think it was awesome. So the third thing I want to do is tell you how awesome you are. You are awesome. And I also want to make sure we're still friends after I just did this to you. I completely flipped the script on you. Are we still friends?
Yeah, absolutely. I really hope that I've been able to answer the questions of the audience. It was a pleasure. I would have loved to see them, but yeah, there were some of the learnings that I thought it would be good to have a chance to exchange and share. Maybe next time with another presenter.
As long as we're still friends, Dr. Goudarzi, there will be a next time.
For sure.
All right. Well, thank you so much for joining us. We will do it again. That is all the time we have for today. We will be back soon. If you're into RNA therapeutics, please join us on September 13th at 11 a.m. when we'll convene another expert panel discussing recent advances in mRNA manufacturing. If you'd like to revisit this discussion with Dr. Goudarzi on demand, it will be available within 24 hours or so at the same link you used today. While you're waiting for that, visit bioprocessonline.com for fresh daily bioprocessing insight and opinion. Subscribe to the Business of Biotech podcast wherever you listen to them.